We are joined today by two special guests. The first one is Professor Ian Constable, who is the founder and currently the patron of the Lions Eye Institute. Professor Constable is a specialist clinician in ophthalmology who continues to practice today. He also has an interest in research and a subspecialty interest in genetic medicine, so his skill set in the research domain is highly relevant to what PYC is doing. And somewhat uniquely, he also has a very deep understanding of the commercial elements of running a biotechnology, drug discovery and drug development company. So he really brings quite a unique perspective on PYC. I would like to say to you that he's an independent source of knowledge for all of you today, but he has joined PYC's Scientific Advisory Board and also the Safety Review Committee of our VP001 program. So he's a semi-independent source of knowledge. And our second special guest speaker is Sri Mudumba. Sri, as many of you are aware, is our Chief of R&D. He has been with the company for the past 18 months. He leads our US office, and I think it's fair to say in large part our Australian office as well. Sri's background before he joined PYC was that he was the head of modality innovation at the world's largest specialist ophthalmology company, Santen. So he also brings a deep understanding not just of the technical but the commercial elements of running a life sciences company as well. We are just getting set up. We'll have the materials back up momentarily for you to refer to, but I will give some overarching context in relation to where today's conversation is situated on the PYC journey. We're having a very defined scope discussion today so that we can go deeper on some of the elements associated with our retinitis pigmentosa type 11 asset, and we are consciously not delving into the wider activities of the company in this conversation. But what you will find, and I think to give you a quick update on where things are at more generally, there will be an increasing focus on other assets in the PYC pipeline as we move forward. Specifically, the company is undergoing a whole of organisational transformation. Again, such seems to be the way in the life of biotechnology companies, but we are moving from a clinical stage multi-asset company to a multi-clinical stage asset company through the course of the first half of next year as our ADOA program progresses into clinical studies. We've got a very, very important readout coming in the imminent future in the ADOA program that we will talk more about in the very near future. Some results in non-human primates that we are hoping to link across to our patient-derived model. So stay tuned for what's going on there. And in the second half of next year, we will also change in terms of the stage of clinical development that we are in. We will move from an early stage clinical company to a mid to late stage clinical company as we kick off our phase 2 potentially registrational study in RP11 scheduled for July. So there's a lot going on at PYC, but I want to bring your focus back to the VP001 program, and what we are going to do is to throw to Professor Constable, who's going to give first an overview of his involvement with the program over the course of the last couple of years as he has seen it progress into clinical development. And then Sri is going to take us through a look back at the achievements of Q3, an evaluation of the current state, exactly where are we in the single ascending dose study, and then guide a conversation in relation to the path forward and the two key readouts or two key milestones that I think investors are very keen to see. When are we going to see some insights on efficacy in that trial, and also what is the timeline in terms of the path to market and revenue generation in that context? So we'll have a look at those, and then we'll open up for Q&A at the end, and that will be an open forum discussion. You can direct your questions to either Sri, myself, or Professor Constable at the end of it. But I would encourage you to try and get maximum value out of our today and to give me a break. So with that, Prof. Constable, I will hand over to you, maybe just to share some overarching thoughts in relation to the journey so far.
Well, thank you, Rowan. I've had a long career managing retinal disease, as Rowan alluded to. I've also overseen 3 successful research through clinical trials to exits in the United States, 1 listing on NASDAQ and 2 sales to major trade entities. So I'm very familiar with the process that PYC is going through. In my retinal life, inherited retinal dystrophies remain a really major unmet need because they cause problems in childhood, and most people are blind by middle age. So it has a particularly poignant effect on the individuals and on families. There are over 250 different gene defects causing these diseases, and only one, a specific rare disease called RPE50, RPE 25 is approved for therapy. Only one specific gene defect, and that requires a virus to transmit the RNA product into the cells. PYC, through previous Chief Scientific Officer, Professor Sue Fletcher, have designed and now successfully proven in a wide range of preclinical studies, which I've studied, an RNA protein conjugate that works independent of the specific gene defect on autosomal dominant diseases. This is a particularly important concept because it's gene agnostic. That is, it could be applied to any autosomal dominant disease, and therefore PYC has an extraordinary platform on which to build if it can prove up the first 2 or 3 indications. They chose autosomal dominant retinitis pigmentosa as the first case. There are 400 or 500 of these people in Australia. Most of them go blind in their 40s. They begin to get symptoms in childhood. The eye is good for this sort of therapy, a simple injection in the eye. And importantly, you can do very precise monitoring, as Shri will show us later on in this talk. Why is it not specific for the gene that's defective? Well, in autosomal dominant conditions, of course, you have 2 genes and one is still working normally. However, it's modulated by a gene silencer. Another gene's producing a protein which keeps the amount of the protein that the target gene produces down to a low level. If you silence the gene silencer, then the good gene can upregulate and produce more protein, and that's what's key to the strategy employed by PYC. It's really the only company, to my knowledge, despite a very large number of gene therapy companies in startup mode doing early clinical trials. It's really the only one that has a synthetic RNA protein conjugate that works in this way, and the IP looks very solid. I think the program execution up until now, in the 2 years I've been looking at the data, has been excellent. The team is really good. Sue Fletcher already went through successfully to FDA approval and commercialization of an RNA product for muscular dystrophy. So this mob know what they're doing, in my opinion, and that is key to progressing the whole technology. The clinical trials have already started, and it's important to note that they're on time and they're on budget.
I think we just lost you at the end there, Prof. Constable, but thank you very much for those introductory comments. I might hand over, Dre, now to you to take us forward.
Good morning, everyone. I think we're going to be talking about some, looking back what we have accomplished in the last quarter, and then going to the exciting stuff, what's going to be the forward view for the program of ARC11. In the last quarter, we have received FDA fast track designation. I will go over the details and what it means to the program and company. And we also have finished repeat dose toxicity studies in 2 animals, both rabbits and monkeys, which basically enables us to go do repeat dose efficacy studies or safety studies in human. And then we have also finished from a clinical perspective, the first cohort of the clinical study, SAD study, we call it single ascending dose study. And we got a approval to go to the second cohort by the safety review committee, which is independent committee. And we have started screening patients for the second cohort. So we're on track for the clinical program. And the more exciting part is the forward-looking forward view for the program. I'll walk through in details what our plans are to go into multiple dose or repeat dose safety and efficacy studies in human, and how we're going to use that to basically find a path to the market. Can I use that?
I think we're better using this one. Oh, hang on. No. Can you use that one? No, it's just on the.
Yeah. From a repeat dose tox studies, as I have mentioned, we have established repeat dose toxicity, and there's no adverse events in both rabbits and monkeys. And we're able to get up to 50 microgram dose, giving a repeat dose, and not seeing any adverse events in the monkeys, which is more relevant to human. What it means is, with the 50 microgram dose, if you're able to get that kind of clean safety data in monkeys, we can go up to 100 micrograms in human. So we know how high we can go in the repeat dose human studies. That enables us there. And then we also have finished the genotox studies in animals, in vivo genotox studies, but the battery of these two tox studies basically enables us to go into repeat dose, multiple ascending dose studies in human. So that part is done. And we got an FDA fast track designation, which basically means it has two benefits. The immediate benefit for this program is we can have frequency of interactions with FDA compared to a regular program, a standard program. Typically they give 3 to 4 meetings to meet with FDA to clarify your doubts or to have a blessing on your design. But in this program, when we have fast track designation, we can have frequent, more frequent interactions by email, by picking up the phone, or even meeting more frequently to get sanity, check on our designs, and then have an agreement on some of the endpoints we're measuring. So it's a benefit to a small company like this to quickly move forward and have this frequency of interactions, which also helps us taking FDA along with us in the path. So they have fresh memory of our program. The second benefit that we get is that's a long-term benefit. When we finished our pivotal studies, we have a complete, good package of clinical efficacy and safety. We can have a typical review time is 1 year to get the approval. We can cut it down to half a year, accelerated pathway, basically. And in addition to that, when we file NDA, we have to file quality and the clinical efficacy safety data. So quality basically means having the product in quality, good quality and robustness and everything. So we have that ready even before we file NDA during the clinical program time. So when we file NDA, we can give them in piecemeal kind of situation where typically we give everything. They have to review all the documents, but we can give, since the quality is in good shape, we can submit quality right away and then move on as we prepare in the clinical efficacy safety data reports. They can finish the quality part of it and then we can provide the second part. So there are some, allowances with the fast track designation for sure. That's going to be really helpful for this program. And with perspective to the human studies, as I just mentioned, we finished the first cohort, which is 3 micrograms. It's well clean, and safety review committee, approved us to go to the 10 microgram dosing, second cohort. I can talk a little bit more about it in the second, next slides. And with this kind of progress and being on track, we see that we have provided the protocol to FDA that we will do 3 doses, 3 micrograms, 10 micrograms, and 30 micrograms. So with 3 cohorts, all these 3 cohorts, we can finish by end of 2023. So basically we're on track for the program. So now I'll go through the next level, the SAD study, a little bit more detail here. So as I said, we have 3 microgram dosing, which is finished. Now we are at the 10 microgram, cohort. And the good thing is, the first cohort took a little long to have the sites up and running and have the patients lined up and the measures in place and everything is validated and everything. So we kind of have a good jump start with the first cohort. And second cohort, as soon as we got the go from safety review committee, we're already able to screen one patient. And second patient is going to be screened next Monday, and both the patients will be dosed week of 16th. So hopefully we will finish the second cohort very quick. And by the time we finish the second cohort, we're already prepping the screening for the third cohort. These patients are available for us through the natural history study that we have started last year. So we have access to the patients, we have the sites now up and running, and we have the path defined. So the completion of these 3 cohorts, I have no doubts will finish by end of the 2023. So what we're going to be doing right after we get the safety of second cohort, 2 things we're going to be doing is we're going to file for dose escalation. Until now, we are only planning for 3, 10, and 30 micrograms. So we're going to file an amendment to test 90 micrograms in human. As I mentioned in the earlier study, we're able to see 50 micrograms safety repeatedly in monkeys that can translate to 100 micrograms. So that enables us to go to 90 microgram dosing in human. So we'll file for that amendment as well. In addition to this amendment, the additional thing that we will be doing is we'll also file an amendment to do multi-ascending dose, repeat dose safety and efficacy studies in human. I'll go through that a little bit more in detail. So we'll file those protocol amendments right after seeing the safety in the second dose, so we can actually follow on and move quickly on the program. So right now we are just assessing the safety assessments in the patients. While we are doing safety assessment, I'll go more in detail about all the measures we are doing, endpoints. Are we picking any efficacy signals? The goal is by the time we finish the multiple ascending dose, we want to pick a good efficacy conviction so we can go into pivotal trial. So this slide, I'll walk through this a little bit more. This is a strategic slide, basically. If you look on the top, we have 4 quadrants in that, in that phase 1 SAD, which is safety ascending dose, safety study we're doing. Each quadrant is 1 cohort. You have 3 cohorts there. We are in the second cohort. As I was mentioning, after finishing the second cohort, safety is done. We file an amendment to the FDA. One of the amendments is to do the 4th dose, which is 90 micrograms. You see the last part. And second is to start a phase 1B open label multi-ascending dose study. In this study, we will start assessing safety by giving repeated doses. We are not a gene therapy one and done kind. We need to keep giving the doses at different intervals to the patients to maintain the efficacy over the long term. For that, we need to establish safety to give multiple doses as well. So that's what we will do in this study. But besides that, since we are going to be doing only one eye treatment, there's going to be one eye which is untreated in the patient. So we'll start looking for the efficacy signal in the treated eye in comparison with the untreated eye. So those are the subtle differences we'll start picking up in the patient's eyes. So when we do that, if we see there's one aggressive path, scenario 1, and there is a little conservative path, scenario 2. And we're pushing forward to see if we can go on the scenario 1 portion. What we will do is, when we do the phase 1 open label study, in the middle, after 6 months or 1 year, after 2 doses, 3 doses, if we start picking up some efficacy signals, there are 2 options we'll have. We'll work with FDA. This is where the fast track will come very handy. We'll work with them to see if we can flip over and turn this phase 1B open label into a phase 2 double-masked, controlled double-masked study, which can eventually become a registration study. That's the one possibility. Or we can have a parallel phase 2 or phase 2 conversion to phase 3, a mass study. That will be used, the data from that can be used for approval, basically. So we will see. If we can convert it, that's awesome. So we don't have to do multiple studies. If we can't convert, at minimum scenario, we can at least do a parallel study while the open label study is going on. So something that happens good here is from the day 1, from the cohort 1, we'll start accumulating safety in these patients with multiple doses, single doses, open label, closed label. We need to accumulate a lot of safety data by the time we provide NDA approval for the to the FDA. That all comes very handy. So the second scenario is a little bit more conservative. We continue to do this open label phase 1B, comparing the treated eye versus untreated eye. The efficacy signals will start picking. If it takes a little longer, we'll complete the study and then we'll do a phase 3 study. That will, if we do an aggressive path, we can almost get a year. That's what we'll push forward and look forward to. And if you do a control, more conservative, it'll be a year later. So we have 2 scenarios there where we can go for a drug approval. Now I'll go to a little bit more details on, so in terms of timing, that's the strategy. We are in 2023. We are where we are right now in the fourth quarter. Hopefully, we can start that phase 2 that can convert into registrational phase 3 study sometime in 2024. In the middle of the 2024 is what we're hoping for. This is an aggressive path. And when you do the study is going to look like, one is a sham control, and then 2 doses we have to give. You have to show a good difference of treatment with the doses that you gave versus sham in a statistical significant manner. That we'll all figure out how to do the design. So now I'll walk through more details on how are we going to pick this efficacy signal. This is where a lot of science and creativity and then experience, everything spills off. We have access to world experts in our retina, like including Dr. Constable. We have Mark Pennesi and David Birch and all the people who have been working and doing clinical research in retinitis pigmentosa and inherited retinal diseases. They know very well, and we have regulatory expertise helping us as consultants to work with FDA and find out the right endpoints. But I'll try to make it a simple version here, how we go about these endpoints, what it means. So on the right-hand side, if you look at the picture, that's the degeneration of sight in an RP11 patient. At the top, you see a clear vision at the early ages of life, and patients start losing the peripheral vision and end up with a tunnel vision, and over the time, they become legally blind over the different decades of life, basically. We're talking about 6 years to 46 years, 4 decades. So it's a slow progressing disease. Some of these factors are disappearing over the time. So the goal is to see in the progression of the disease, which patient is coming into the trial and which factors are the ones which we need to focus on, and how our treatment is going to influence on those factors. That's what it is. But then when you work with the regulators like FDA or EMA, 2 things they look at is either visual function or the functional vision. Is it getting better? Because that's what makes sense to the patient or the physician. So those 2 are the final goals, that we need to improve the functional vision or the visual function. Now we get down to see what are the markers that will help us to monitor those functional vision or visual function. So if you look at on the right-hand side, we have visual acuity charts, basically reading the numbers of letters. The typical standard by FDA is you need to have 3 lines of improvement of letters reading, which is basically talking about 15 letters of improvement to get an approval, which is going to be very difficult in this disease, which we call best corrected visual acuity, which is the case in macular degeneration, wet macular degeneration. That's the endpoint FDA buys into, and then there are approvals. In our case, the important one is going to be low luminescence visual acuity, the bottom one, LLVA we call it. With the dim light, how many letters is the patient able to read, and with the treatment, is the patient going to improve the number of letters? That's what we want to pick. And which are the patients who's going to be helping us do that? Patients who are in the borderline of losing the peripheral vision. The immediate impact of retinitis pigmentosa 11 is people start, becoming night blind. They start knocking things down in the nighttime. They can't read, they can't see things. So we want to pick that signal. There are going to be patients coming in that zone of window, and when they walk in, and we want to monitor those things in them. We are monitoring all these parameters, but those patients will be qualifying for us to test the change in the low luminescence visual acuity. Similarly, visual field is basically being able to see fields of vision. How far can you see peripheral vision? What is your ability to see? I have an example here. We do multiple, we call perimetries. We do macroperimetry, static perimetry, we do kinetic perimetry, different visions, basically to understand what is the visual field level of patient's ability to read or see the signals. And this is an example here. I have a healthy patient, 65, 70-year-old, and you see nice vision, peripheral vision and central vision here. And you have, these are the numbers of decibels, which is intensity of signal. They are able to see. The higher the number, the better the vision is. You see multiples of this, visual, fields here. And when you look at the patient, they lose all the peripheral vision. This is the real patient's data, that you can access. And you see all these dark signals are basically, there are no intensity they are able to read. Only the central vision is there. So these are the people somewhere here, the ones who are in the tunnel vision. So we want to pick these patients and if there is this periphery here where there is some level of visual field effect, we want to preserve it. We want to make it better. What we are trying to do with our drug is to improve the health of photoreceptors by increasing the protein upregulation of particular protein called PRPF31. So we want to be able to upregulate it and then improve the health of these photoreceptors so their sensitivity get better and better. And so you can preserve the vision, whatever the shape they are in. So we will be getting some of those patients who are in that situation and that quota basically. And since we know that the drug is, when we tested in vitro in patient models, we could see that the drug is able to upregulate the necessary protein PRPF31 and improve the health and morphology of the cells. We want to be able to see those markers by imaging. They are structural markers. So we use these two markers to see SDOCT. What it tells is there is photoreceptor zone, we call it. The density of photoreceptors, we call it ellipsoidal zone. Is it increasing or decreasing as what's happening with the treatment? So if the density is maintained or increased, you are actually helping the health of photoreceptors. And now you can infer that to improvement in sensitivity, improvement in visual function. So we're going to be measuring that as well. And FAF is another, which is fundus autofluorescence, which is another structural marker we're going to be measuring, where you start seeing all the patches in the peripheral vision because the photoreceptors are degenerating. As you improve the vision, as you improve the function of these photoreceptors or their health, they start glowing better with the fluorescence, and you can start picking up those differences. And we want to pick those differences. These are the ones which are going to help us to see whether or not drug is improving the efficacy signal. And that's what it gives us the clinical conviction, basically. So in regards to visual fields, one is perimetry we are measuring. These perimetries are standard. They've been there in the ophthalmology field for several years. But the one approval that we have with the gene therapy for RPE65 that Dr. Constable was mentioning, Luxturna product, they got approval based on a mobility test. That's an example here. Basically, there is, they make a maze with lots of hurdles here and there, objects, and they keep changing luminance, and the patient walks through those mazes. And if the patient is knocking down these objectives, the peripheral vision is bad. And with the treatment, if the patient is eventually able to walk without knocking down some of these objects, you show the difference in this, ability to walk. It's a real functional test. So that's another test we are actually measuring in these studies, both, SAD and MAD studies. So when we do all these things, we will start collecting the information on which kind of patients are responding to what factors well, right? And then we also are engaging the regulatory authorities to see an example. They were only based on functional vision, visual fields, or visual function, BCVA or LLVA. If you want to work on only perimetry and the visual charts, it takes a long time to show the difference. But FDA has been progressive, and they've been working with the companies and the research, and they have approved a couple of endpoints. An example, FAF, fundus autofluorescence, for geographic atrophy due to macular degeneration. They could see the difference in diameter of the hole that is happening over the time with the atrophy. You are able to arrest the progression of the diameter of the hole, then you get an approval. That's a structural endpoint. FDA was, able to give that endpoint to them. They got approval. So we're going to be, latching onto that as well. And SDOCT, there's another trial that's going on for MACTEL. Again, FDA is amenable to the endpoint where they were able to show the difference in the ellipsoidal zone photoreceptor density changes over the years. They were able to use that as an endpoint. So we're going to be catching on those two endpoints. And maze test is something that has already been approved for Luxturna for gene therapy. But then when you go back and retrospectively do analysis, with the, a lot of people have looked into it, there are a couple of upstream signals that you can pick in the photoreceptors, which are correlating with these markers that you're measuring in the clinic. Example is ERGs, which are electrophysiological, techniques that people use. But another one, which is more important, is FST, full field stimulus threshold. These two will give us a good understanding of sensitivity of photoreceptors. Improve the cell health, you're improving the morphology and you're improving the function. That will improve the sensitivity of the photoreceptors. That eventually will improve the visual function of the person. So there was a good correlation between this maze test improvement and FST measurements. So we are actually measuring FST as well in this study. In addition to all these things, eventually when we go to approval of the drug and when we want to get the pricing for the product in the United States, in addition to the markers and the approval that you get from the FDA, they are looking at the patient-reported outcomes, because eventually what does it mean to the patient? And that's what is going to help us really put, when we go to CMS, basically Center for Medicare Services, our payers, besides the approval is one thing, FDA is good, and you're getting approval, but what does it mean to the patient? If you show the outcome differences, that actually will help them, reimbursement process. So we're also collecting those information in these trials, patient-reported outcomes, basically, all those, how they are impacted by these, changes. So all these things are going to be, measured in this study. And our idea right now is to go forward when we go to the pivotal trial to use a composite endpoint, because when we have the entry of the patients from different, different, disease statuses, whether early stage or mid stage or late stage, they're all going to be responding differently for some of these, functions that we're measuring. So we're going to set a composite endpoint saying that either a change in LLVA with 5 letters or 10 letters, or a change in perimetry with 5 decibels or 10 decibels, or a change in SDOCT with density of photoreceptors, or change in the sensitivity from minus 10 decibels to or minus 20 decibels to minus 30 decibels, is going to be seen as a significant difference in the patient, which is clinically meaningful. So we're going to design the study by putting this either or, we call it composite endpoint, such as the only challenge that will be there is for each one of them, you need to have a small amount of population. For LLVA, if you're using, you need to have at least 3 patients or 6 patients. So we have a pool of patients. That's what the natural history study is helping us. We have, about 30 patients enrolled in natural history study. We'll continue to enroll, and we can put them into these programs, and they all will come in with different presentation of the disease, and they will respond to one of these parameters, and we'll collect all the endpoints as a collective endpoint to get the approval. And that's my explanation. Dr. Constable can chip in for any of the endpoints that I talked about.
Thanks, Sri. One of the interesting points from a commercial point of view or approval point of view is that all these patients are known. Because it's such a demanding disease, it's a horrible disease, they all collect in specialist centres. So in every city in Australia, and certainly in every place that has a developed health system, the cohort already exists. So once approval were to occur for a new therapy, treatment would roll out without a marketing program. It would just roll out to everybody that's on the books if FDA approved it. So it's an attractive area in the sense that the orphan pricing is already established for many rare diseases, typically in the US, $150,000 per patient. The rapid uptake towards peak sales would occur very quickly.
I think that's a very salient point, Prof. Constable, and thank you. The other dimension there, and I think you hear the challenge that PYC has, is that this is a slowly progressive disease. So we have an opportunity, but also a challenge to try and understand how is the disease progressing in the absence of treatment, but also how is the signal going to manifest in what is essentially a heterogeneous patient population. We think of them all as having RP11, but different patients at different stages of the disease have different implications of it. So you heard Sri speak about the loss of the peripheral vision in the early stage patients, but the loss of the central vision in the late stage patients. The additional big commercial opportunity that presents to us is, in the event that we can find that efficacy signal, you capture almost the entirety of the RP11 patient population, because they all still want treatment, because they all have some part of their vision left. Even those who are very late in the disease course place a very high value on the last few percentage points of their central vision. So there are some challenges in the clinical determination of the efficacy signal, but also some opportunities that represent the flip side of that same coin. And I think the rapid market uptake and the known and captive patient population, as Professor Constable points out, is quite relevant in that context. Look, they say nothing ever goes to plan in drug development. It doesn't usually extend to a failure of the lectern microphone, but nor your head of non-clinical development dialing into the meeting and getting feedback partway through to overcome any challenge that is thrown at us at PYC and still deliver outcomes. So hopefully you've managed to bear with us through that technical failures. And we're going to open up now for Q&A, or an opportunity for people to ask questions. And you're more than welcome, as I suggested at the outset, to direct them either to Shri, to Prof Constable, or myself. We can go beyond the scope of the RP11 program, but we would really like for you to get as much as you possibly can out of the pathway in RP11, the path to market. I know there's a lot of interest in this respect. So if we can keep them ideally focused there in the first instance, that would be very helpful. I don't know, Shri or Prof Constable, if you had any final comments before we transition to Q&A?
No, I think that's fine.
Okay. We have got can we explain at what dosing you start to see efficacy? Are these early low dosing studies showing any results, or is it purely for safety? Shri, do you want to address that one? You might be best standing near the microphone.
The early doses are basically meant for safety. We're going to build it up as 3. And if you look at they are like semi-log improvements, 3 to 10 micrograms. So, and then also depends on the kind of patient who is coming in. If we end up patient who has very less scope for improvement, it's very hard to see with one dose any efficacy signals. But it's predominantly for safety. And if you get lucky, the patient is with a manifestation that can be improved very quickly, and they get into trial, maybe we'll pick some efficacy signals by 10, 30 microgram doses. So that's very forward thinking.
Okay, the next question, how does the work that PYC are doing differ from that of Opthea, or are they complementary treatments? I know they're entirely different. We're actually pursuing different disease indications to Opthea. So Opthea are pursuing an indication called wet age-related macular degeneration, looking for a differentiated therapy in a condition for which there are already approved therapies. So Eylea and Lucentis are significant therapies available to patients in this domain. The disease indication that PYC is going after doesn't have any treatments available at all. And in fact, the VP001 candidate is the most advanced drug for this particular blinding eye disease called Retinitis Pigmentosa type 11. So they're very different indications, completely different patient populations that we are creating therapies for. The next question is, if the improvement in the eye can be seen in these safety studies, will each person display the same improvements in their eyes, or does it differ from each person what effect the dose will have? Prof Constable, I might send that one across to you, whether or not each patient will respond in a uniform fashion, or whether we're likely to see different things in different patients.
Very likely to see different things in different patients, because the enrolment will include a wide range of degree of progression. As Rowan pointed out, even people who have advanced disease are going to want to try and go into trials, because if it works, it will perhaps preserve their very residual vision, whereas other people may have quite good vision, albeit with a decreased visual field and loss of night vision. And they will have different signals. So it's not going to affect the whole cohort the same way, which is why Shri has designed so many different battery of tests, which will pick up some elements of that in the earlies versus the middle progression versus the late stage disease.
Yeah, I think that's a nice answer, and touches on a related point just in terms of the fast track designation that Shri spoke about earlier as well. What we have here is a clear recognition from the FDA, the most important regulator from PYC's perspective, that this is an important potential new therapy for patients with RP11. And we'd love to give you the colour on exactly what the path to market looks like, but we can't do that yet because there are input variables that will inform part of the answer to that question that we don't have. So we've got a very clear understanding of what we need to do through the remainder of 2023 and into 2024 as we transition towards these registrational studies. But the information that we really need to fully design those registrational studies and understand how long they need to go for, how many patients need to be in them, which endpoints we're looking at, come from the natural history study. So the information that we're gleaning over the course of the disease in the absence of treatment, They also come from the observations that we're making in the interventional study on how patients are responding on the different endpoints that we are assessing. And so what we're doing there, and Prof Constable just touched on it, we're measuring a huge raft of different assessments of visual function. And we are looking for those on which the patients are moving most quickly, so we can use those to inform the design of the clinical trial. The other very important dynamic element is that you see a progressive awareness on the part of the regulator, the FDA in particular, of the importance of these therapies for patients who have unmet needs. So there's a softening of the stance. There was a very important recent publication on precision medicines. It came out about a fortnight ago, where the FDA are saying, where you have a drug that has got a very strong mechanism of action, a very well correlated mechanism of action to correction of the disease, we will accept a lower level of evidence in relation to the approval of that therapy prior to a post-marketing confirmatory study. So that the regulator generally is moving towards being more flexible in relation to looking at the data sets generated in these rare diseases. Although within the FDA, you have different departments, and some of the departments are quicker to move than others. So it's a bit of a movable feast at this point in time, but hopefully we've set out some of the principles that are going to inform the design of those later stage studies. We have a question: what percentage of your patients are sham-controlled surgeries? That one is very quick. In the phase 1, none. We're looking at the sham-control group as we move to the multiple dose study, so 0 in the SAD. If a patient participates in phase 1, will they be eligible for a later phase or perhaps an extension study?
Yeah, that's a very good question. Actually, we've been strategically thinking about it. That I'll give a little long answer, sorry, but we have received fast track designation for this product, which basically mandates us to publicly announce our compassionate use policy for the drug. Right now, because we do not have enough safety evidence, we don't want to give compassionate use as an expanded access to the patients. But in the future, after we finish our second cohort safety done, we're going to open it up to make it available to the patients on a compassionate basis. So what it means is the ones who got 3 microgram dose, after they finish 6 months of safety, we collect their safety information. If they're interested in getting the drug for compassionate use, they can get it. That's one way. In a compassionate use, basically, they can continue to get the medication as long as they see the improvement. And we'll keep collecting the information as supportive evidence for us when we actually go to the approval. And we also establish safety in that process. That's one door. And second is, once they finish their safety for 6 months, they can always jump on the board for the efficacy study when we do the pivotal studies later.
I've got a question actually that I'd like to address to Professor Constable. I'll jump the queue. That's the good thing about controlling the microphone. You can go straight to the front. Prof Constable, we have placed a great deal of stock in 2 particular dimensions of the drug development journey. The first one has been on selecting genetically validated targets. And I was very pleased to see recently a retrospective by Alnylam, another company that focuses on monogenic diseases, coming out and saying that they had a 64% chance of a market entry upon initiation of a phase 1 clinical trial, which is quite a radical number when you consider that the benchmark industry success rate is only around 10%. So that was the first dimension, but the really interesting one is that throughout the course of your career, you have seen the availability of these patient-derived models come to hand. Only really recently in the last 10 years, and the sophistication of them is getting a lot greater in recent years in particular. We think that there is a much higher degree of fidelity in testing human genetic medicines in the context of the human genome, and specifically the particular target cell that has got the deficit in the indication that we are looking at treating. I wonder if you've got any thoughts on the conviction or translating through to the probability of success of medicines in the clinic that have been validated in the context of patient-derived models, as compared to those that may have been validated previously in an animal model that may not directly recapitulate the human disease process.
There's been a lot written about this, and there's no question that the human cells put into 3-dimensional cultures and then with the defect present, and then applying the specific potential therapy has made it so much more precise than the old programs where we used to create animal models. I did huge numbers of the clinical trials for macular degeneration, for wet macular degeneration, and we were making models of blood vessels growing in monkeys' eyes in other countries, and spending a fortune and sacrificing lots of monkeys, whereas now that sort of study is confined to safety, which is still necessary, but it's reduced the cost, it's reduced the time, and it's greatly increased the specificity, having these cell-based three-dimensional models of human disease.
Thanks, Prof. Constable. The next question is, do we expect the treatment to maintain the patient's vision or potentially restore lost vision? I'll throw to you again for that one.
With the kind of approach we have, definitely we will maintain the vision, that's for sure. And restoring the vision is dependent on how many photoreceptor cells are on the verge of death. And then once you start, picking up the protein levels up, you bring them back into their, improvement in their health, and then maintain that health status. In that context, there will be some level of restoration of vision.
Can I jump in there, Rowan? There are two aspects to this, I think, and we don't know until we do studies with concentrations of the therapy that are going to make a difference. The first is the peripheral loss of vision. Seems to occur, at least in the few studies that have been done, and they're small and inadequate, but about 8% a year, the visual field, the side vision, decreases. And it may be that you have to wait a while for that particular signal to show a difference. 2 years, 16% on average. The untreated eye would be worse than the treated eye if you were showing a signal. However, we do know in the models that the cells in the middle that are still functioning in the earlier cases don't look normal, and with the treatments in tissue culture models, those cells turn around and become normal very quickly. And it may be that people with, say, 50% vision with a ring of field that are still relatively early or mid cases, in fact, the cells that are still functioning improve. And it may well be that in that cohort, you get an improvement, and fairly quickly. We don't know.
I think that's a really good point, and it does correlate with what they observed in the Luxturna clinical trial. Patients on the maze that Sri described for you earlier, where they get the patient to navigate through an obstacle course under different luminance or different lighting settings, actually saw an improvement, an ability to navigate the course successfully at lower lighting settings after treatment with the drug than what they had before they started. So some very interesting potential excitement coming there. Will we look to determine optimal dosage based on early, mid, late state? When it goes to market, will this be a variable dosage disease stage prescription matrix? Sri, do you want to take it?
Yeah, we are very likely to have a single dose, marketable dose. That's what we're going to be doing. We are doing 4 doses through these multiple ascending dose studies and single ascending dose studies, but when we actually go to registrational studies, we'll do 2 doses versus sham, and we'll pick the best dose there, optimized dose.
Thanks, Sri. Some of these are a bit quicker than others. Prof. Constable, the next one might be a good one for you. Do both eyes tend to deteriorate at the same rate in RP11?
Yes, that's a quick one. Yes, they do. Inherited retinal dystrophies are noted for their symmetry, all of them. Virtually every patient has a similar degree of progression in both eyes. So the second eye is a great control for the treatment in the first eye.
Terrific, and thank you. The next question is, what other areas outside of the eye are the most exciting for PYC and why? Sri, do you want to me to do it? Or you? All right, well, I think you know that we have got a discovery pipeline that addresses the eye and the central nervous system and also the kidney. And we are very close to an exciting readout in a program that we are hoping to add to the pipeline within the next couple of months. So stay tuned. We have got a pathway to an expedited human readout that will largely inform the prospects of one of those assets moving forward. So we'll keep you remaining tuned until we have confirmation that we can do that. expect to hear more from us in that respect. Is the treatment one dose or is it an ongoing treatment? That's a nice and quick one. This is repeat dose. So the nice thing about the RNA therapies is they are very durable. So they've got a long dosing interval, but they don't make any permanent changes to the genome. So patients will have to have repeat dosing. It is a nice long dosing interval for them, much longer than what we see in patients on the anti-VEGF medications that we spoke about earlier in wet AMD. Is it possible? Oh, sorry, you go.
Which, incidentally, Rowan, is good for a commercial entity. If you have to do multiple doses, that's far better than a single dose.
I told you he had a unique perspective that spanned all three domains of clinician, researcher and commercial dimensions. It's also very good for the patients because remember the PYC strategy is we have chosen indications where only an RNA therapy can succeed. You need to have a retention of the endogenous control of protein expression by the cell. Too much of the protein as well as too little is going to manifest in a disease phenotype, which is why we are kept very clear from a competitive landscape perspective from other modalities. So an important point across both fronts there. Is it possible that the untreated eye, which is being used as a control, may also register some efficacy independent of the treated eye being treated with the drug? Could this affect the success of the trial? Street, want to take that one with special comments from Prof?
Definitely, yes. Yeah, I've been through a lot of ophthalmology programs and done these intravitreal studies for the last 15, 20 years. The kind of modality we chose, when it is a small molecule, when you give intravitreal injection, the likelihood of getting into systemic circulation, and one of the reasons we are doing intravitreal is the drug is not going to reach to the eye if you give intravenous. So when you give in one eye, when it gets into systemic circulation a little bit, it doesn't really necessarily go to the next eye, the fellow eye. So that the question is very, loaded, I would say, because this has been only one incidence. All the small molecules, the modality that we're dealing is not a problem at all. There's one single eye you treat, and that is the eye which is going to get the drug. I think there was some evidence of gene therapy. When you give vectors in the eye, they have the ability to go into the fellow eye. And very fortunately, that's not what we are doing. We are doing a small molecule modality. We don't have that issue.
Perfect. We'll try and move through these last few quite quickly. Will we be seeking from the FDA orphan drug designation, pediatric rare disease designation? Can we advise of timing? The answer is yes, we will be seeking both. So we know that it's an orphan indication. And we also know that the median age of onset of patient symptoms is 17 in RP11. So that should mean that it's eligible for the pediatric rare disease designation as well. Do you want to touch quickly on the timing for that?
Yeah, I think once we establish the safety in the adult population, then we will start rolling it down to lower age, and that's about the time that we can apply for pediatric designation. And orphan designation sometime in 2024, we'll definitely apply for it with any early evidence data that we get in safety study, plus a lot of robust patient data that we have together will help us get the orphan designation.
We've got one for you, Prof. Constable. If he is aware of the upcoming ADOA data, could Prof. Constable comment on whether he shares the view that the imminent data release is as important as Rowan? Oh, that's not very favourable. Made it sound at the beginning of the presentation. Prof, maybe to give you some background there in relation to what we're waiting for is the fair context for answering the question. What we are looking to do here is to link the answers that we can only glean from animal studies that relate to following the drug from the tip of the needle to the target cells that are affected in the disease. We need the complexity of a living system there. We need to know whether or not the drug is safe and well tolerated in the context of a living organism. We need to know how much of the drug can survive the vitreous, which is quite a hostile environment, traffic to the back of the eye, cross the inner limiting membrane and enter the retina. That's a critical piece of information for us to establish through the retinal concentration of the drug, so the amount of drug that we are able to safely administer or achieve in the context of a non-human primate, a monkey. We then transition across to the patient-derived models that we spoke about beforehand. So we use the same concentration of drug in the patient-derived models. As a reminder, these are patient-derived models that are gleaned from patients who have the disease, so they have the genetic mutation that we're looking to treat. You send those cells backwards to a stem cell, and then you send them forwards to a retinal ganglion cell, or the type of cell that is specifically affected in the particular indication that we're looking at, in this case ADOA. We are hopeful, it's not guaranteed that it is going to work, but we are hopeful that we may actually be able to see OPA1 gene modulation in the non-human primate as well. But the non-human primate OPA1 gene is similar enough to the human gene, and the drug is obviously a human medicine, that we can actually see an increase in the expression of the OPA1 protein in the non-human primate, because then you don't need to go across to the patient-derived models at the level of the concentration of the drug in the retina. You can actually look at the ability of the drug to modulate the target gene expression inside the target cell, inside the target tissue, inside the non-human primate. And that is a very, very powerful link indeed. So, Prof, that's what I was referring to with respect to the data that we are waiting for and is imminent. If you can share any perspectives that you have on the ability to link between a wild-type monkey, non-human primate, and patient-derived models, that signal, knowing that we have got full functional rescue in the context of the iPSC IGCs when we increase OPA1 expression.
We'll just have to wait and see. I don't know, but in general, I think it's a good indication. I think that it's another unmet need. Optic atrophy in dominant families is often severe. It's not always severe, but it's often severe, and there will be a very significant group of patients wanting treatment if we can get it there.
I think the question is directed towards your level of enthusiasm for the non-clinical data pack in the event that we can establish those dimensions that I spoke about. So a PK profile that suggests a single-digit micromolar concentration in the non-human primate retina, and ideally demonstrates that we can modulate OPA1 gene expression, so increased OPA1 protein. If we had that and we were able to link across at the level of the OPA1 upregulation or the concentration of the drug administered to the patient-derived models, where you can see an increase in mitochondrial ATP production, an increase in oxygen consumption on OCR for those cells, and ideally a greater resistance to apoptosis, how would that inform your conviction in that molecule as it moved into the clinic? And is there anything else that you would like to see in that non-clinical data pack as that molecule moves into patients?
I'd be enthusiastic about that, but I don't think I have to worry, because FDA will be looking at that and they will make their decision on it. But I would bet that they'll approve human trials on that data.
It's a cautious yes, I'm going to ascribe that one down as. Next question. If every single hurdle was cleared on time and results were at the best spectrum, what date would the first commercial dollars be banked? Really nice to have you to throw to here. Drew, would you like to step up and answer that question?
So I think, assuming we start sometime, next year or following year, the pivotal trial, we will have to monitor for almost 2 years to show the efficacy difference, the impact of the treatment on the patient, which basically means, treatments like gene therapy, after you treat, you wait for 2 years. In our case, it's a repeat dose treatment, so we'll be continuing to treat in those 2 years. So we'll do like 3 or 4 doses, let's say every 3 months until 9 months, and we give another 3 to 6 months time frame and look at the final read. So we will be a 2-year treatment plus outcome measurement kind of study. So it will be done within 2 to 2.5 years, and after that we file for NDA, and as I said, with the fast track designation, we should be able to get in 6 to 8 months approval. And now we're talking about, a commercial launch. So I didn't give you the date exact, but I gave you the time perspective.
I think you can work it out from there. And I think that's quite reasonable. Based on what we know today, we don't have a definitive natural history study, but we do have some anecdotal natural history studies. We see around an 8% decline in the peripheral vision of the patients who are at that early to mid stage of the disease. So a 24-month window, 16% decline in vision should be enough to be able to see that delta reliably between the treated and the untreated groups. What possible side effects could stop the study? Constable on the SRC, you can fill that one.
The first patients that have been dosed had no significant side effects whatsoever, but they only had a tiny dose, 3 micrograms.
So what would you be looking for? What do you think are the most likely signs of adverse tolerability that you'll be keeping an eye out for there?
You're putting foreign material into the eye, so inflammation, immune reaction, that would be manifested by maybe swelling of the retina or inflammatory cells, which you can see an image when you're examining these patients every week during the safety studies. You may also see some toxic effect of a molecule on the appearance of the cells. They're all being imaged every week in minute detail with OCT scans and so forth. You'd be unlikely, I think, to see functional defects, but if somebody had some vision and they'd lost a bit, that would be an adverse effect, and you'd want to know why that was so.
Yeah, that's helpful, and thanks. I think it informs a risk-benefit analysis. So if there's anything that indicates that the therapy is making the patient's vision worse or accelerating the deterioration in their vision, we would be concerned in that context. All right, that exhausts the questions. We had some more that were submitted in writing. Carol, have we thematically addressed them, or are there?
A lot of it was just when's FDA approval, and basically the steps that we've taken to minimise any risk in delay for the approval, which we've kind of gone through.
Yeah, I think we've largely touched on that. Actually, I don't know that we did speak about it in the context of the presentation, but we have added a second site in the US now has ethics approval. Oregon has come online as well. They have screened patients actually for patient cohort 2, and we are looking at a streamlined and efficient execution of that second patient cohort. So we remain very much on track for execution of cohort 2 and also cohort 3 before the end of this year. And we have opened up some optionality in relation to the different sites that patients can attend in the US, which is encouraging. We're still working hard to get Australia online and trying to achieve that before the multiple dose study kicks off in July next year. So generally, I think we, just moving back up now to the overarching context for PYC, we have a stated ambition of 3 first-in-class and potentially disease-modifying drugs into human development before the end of next year. So it's a very lofty ambition. As a company, we are continuing to invest in scaling our platform, not just because we have important unmet patient needs, but also because RNA therapies are very much establishing themselves as a major modality in the context of a downturn in the biotech market. So when we see things turn, and they will turn eventually, PYC has made a significant investment in scaling our position into the tailwind when it eventually arrives. So it hasn't been a good time in life sciences, but we are very much buoyed by the quality of the data that we are generating, and that informs everything in the downstream environment here. We know that nothing has changed in relation to the ultimate value of these assets and the impact that they can have on patient lives. So if and when things do turn, expect to see PYC turn quite aggressively into that. We also are making clear and unambiguous progress, as Prof. Constable mentioned before, in relation to critical milestones that are changing the intrinsic valuation of these assets. We are removing risk and therefore adding value to the assets as they move forward. In the context of precision medicines, a lot of that risk is actually taken out in the non-clinical setting. I've been at pains to try and describe to people the shift in the risk curve. If we are starting clinical trials and we're anywhere near the industry figures that are quoted, the 50, 60, 70% P success in the context of the clinic, that means most of the risk actually had to come out in the non-clinical setting. And you've got 2 very important milestones. One we've just spoken about in relation to the ADOA program that is coming out in the very near future, and another one where we are looking to add a 4th program to our pipeline in a very large commercial opportunity, one that will dwarf the RP11 and ADOA programs in the event that we are successful. We're looking to achieve that before the end of this year as well, with a view that we will accelerate that program into clinical development next year in the event that we're successful. The PMS program, the Phelan-McDermid program, has got a slightly more complicated path through the GLP toxicology studies because we're looking to include a pediatric patient population in the clinical trials there. So that one will likely move into 2025. But what the company is committing to doing here is putting at least 1 first-in-class precision medicine into clinical development every year from now on, which means that we are now the beneficiaries of a very exciting and steady flow of news that is going to move the needle for investors in this space. So that's the overarching context. We thank very much Professor Constable and Sri for their contributions. I don't know, Prof. Constable, did you have any final thoughts that you wanted to share?
No, I think you've summarised it well.
Thanks very much for your time and ongoing support and commitment to the program. We very much enjoyed your engagement, Prof. Constable. We look forward to touching base with you again, hopefully in an investor-facing forum. Sri, anything that you wanted to add?
I just want to add one last point is, when it comes to commercialization of the product, typically when you do your pivotal trials, if you take a set of patients and when you get the data out, the label of the product is going to be usually representative of the set of patients that you tested. One advantage we have in this trial is then going with the composite endpoint is we're taking patients of all different disease presentation and putting them to different markers. So when you get the label, that's going to be for the patients all different stages of RP11. That's going to be a bigger, wider range of opportunity, basically.
That's the nice thing about being a company that is directed towards precision therapies that address the underlying cause of the disease, is that you have very high conviction that you're actually going to change the lives of all of those patients with the specific indication. So, look, thanks very much, everyone. Thank you for bearing with us through the course of the technical difficulties that we experienced earlier on, and hopefully that's given you a good deal of colour in relation to where we're program specifically. We'll be back in touch with you very shortly in relation to helping you understand the implication of the readouts that are coming in the very near term. They're very exciting for us as an organisation. We look forward to sharing them with you. Thank you.